I wanted to get a discussion going about the major forward jump in sequencing that's been occurring that was published in Nature in November 2008. There were 3 papers published as companion papers in one issue of Nature. The first whole genome sequence of an African ancestry individual, a whole genome sequence of an Asian individual, and the genome sequencing of an individual with acute leukemia, along with the normal skin sequencing and further sequencing of the leukemia in relapse. This was an extraordinary paper, that is a leukemia paper, because what it did was through germline DNA sequencing and tumor DNA sequencing, there was a discovery that there was, by at least focusing on coding regions, exons, there were 8 genes that were found to be present at the time of the diagnosis as well as at the relapse, representing a dominant clone. And these 8 genes really gave insight, really for the first time, in individuals' cancer, what would be conceived as the driver mutations. It's a segue potentially for a better therapy, but it demonstrates the power of where sequencing is going. Who would have thought it'd be possible? In fact, whereas it costs in the billions of dollars to do the first whole genome sequencing of several individuals, the Human Genome Project in 2003, and with the Celera Venner project at that time, the costs have now dropped for whole genome sequencing to somewhere less than a couple $100,000, and Complete Genomics has already announced that in the first quarter of 2009, a $5,000 whole genome sequencing will be possible. So we're starting to see a time in which, for example, in cancer, sequencing an individual's tumor and using that individual's germline DNA as background may help to ferret out the responsible driving mutations that are in that particular person are associated with the cancer, demonstrating a new potential capability of designing individualized treatment. So this is exciting. This is just an enormous step forward in a relatively short period of time, and hopefully we'll see continued progress that is in this accelerated stage of the sequencing. So I'd be interested in your views as to whether or not you believe that these represent substantive steps forward, where you see other major caveats and issues regarding this revolution in comprehensive sequencing. So thanks very much for your attention.